So I'd like to introduce Ludwig Geistlinger, who is a postdoc in my lab and a true expert at gene set enrichment analysis, something that he did during his PhD and has continued working on ever since. He's the author of the Enrichment browser package, which allows you to perform enrichment analysis using a variety of different methods in bioconductor and compare the results from all of them. And also the author of a benchmarking package for benchmarking existing methods on data sets of interest, on benchmark data sets that he collected, and also for benchmarking new data sets, a paper that was just published in Briefings in Bioinformatics this year. So it's a great pleasure to pass it off to Ludwig, and I look forward to your presentation. Sorry, let me interrupt and say one more thing. I want to encourage everyone to make use of the BioC 2020 workshop Slack channel. If you have, so use the polls section on Pathable for questions that you would like to be asked of Ludwig at the end of his presentation, and use BioC 2020 workshops for TA type questions. If you are having trouble running something, if you would like some help with trying to run the workshop, or questions that you would like answered in real time, ask them there. And I will be there on Slack. Marcel Ramos and some other people from my lab will be there on the Slack channel to try to answer those kinds of questions right away. Okay, take it away, Ludwig.
Perfect. Thanks, Levi. Share my screen here. I hope you can all see that. Just pointing out that all the materials are available here also in the chat. If you go to the workshop, there in the chat section, I collected all the links that will send you off to the slides, and also like trying out this workshop on these cancer data science instances. All right, it is of course a great pleasure for me to kick off day 2 of the online Bioconductor Main Conference 2020 here with my workshop on one of my favorite topics, which is a gene set enrichment analysis. It's the 4th year in a row that I give this workshop, starting 2017 on the Bioconductor Main Conference. And over these 4 years, we have constantly maintained and improved this workshop, also incorporate recent developments. And every year the workshop has been a little bit different. And of course, given the circumstances, the specific circumstances of this year, also this year's format is quite different to what we used to do. So as I plan it, I will give the first 30 minutes a bit of an theoretical overview when it comes to gene set analysis theory, but then I also give a bit of an orientation on the different workshop contents. I mean, of course, the last years we had a bit more of a looking over the shoulder and hands-on experience. Here I will basically orient you where to look at the materials and try things out. And I also will give a little bit of a demo using one of these cancer data science instances. I reserved 20 minutes in the end for questions, and if we have time, a little bit more of coding examples. So the really great thing, thanks to this huge effort from Sean Davis and Levi, it's really never been so easy to try these things out yourself. And that's not only now, during the time I'm talking, but also after the conference when you likely have more time and also silence to check these things out. And the main address that you likely want to follow here is the website of these workshop materials, and later on also to try these out live. But after the conference, and that's really the great thing, all these materials are living on GitHub, and you can go there, install it on yourself, and basically work in details through things that are maybe more of interest of you. And I really encourage you here also to maybe contact me if there are issues arising either via email or the various Slack channels in our BioC's community Slack. Most of the contents are based on the Enrichment browser Bioconductor package. So if there are questions concerning documentation or specific functions that I introduced today, the bioconductor enrichment browser package is like that you would like to look up for that. All right, so gene set analysis or also gene network analysis is, as I guess all of you know to some extent, an essential step in the standard analysis workflow of high throughput gene expression data as measured with microarrays or RNA-seq. So we're talking here about transcriptomic data, and gene set analysis typically bridges your upstream processing step and gene by gene analysis with the eventual biological interpretation. And the main question really here is when I talk in a differential expression setup, which I will mostly do for the course of this workshop. So we have really differential expression analyzed for all the genes that I have measured in my transcriptomic experiment. And I'm then interested, are there certain enrichments or overrepresentations with known functional group of genes as, for example, defined in the gene ontology or the KEGG pathway database. So there are many methods for conducting gene set enrichment analysis, and a very pre-seminal paper that is likely a good first read when starting to deal with the topic is this paper here from Goman and Willman from 2007, which makes quite fundamental distinctions of concerning the null hypothesis, what gene set, what the different gene set tests analyze. And they basically divide here between competitive tests that basically check whether my gene set of interest contains more differential expression when compared to the background, when compared to all the genes not in the set, when compared to all the genes measured, as opposed to self-contained tests, where I basically analyze this gene set in isolation, not comparing to the background, but basically my null hypothesis here, no gene in the set of interest are differentially expressed. And of course, these theoretical distinctions here also have quite practical implications. I mean, if I look, for example, here at the competitive one, it becomes clear that this is a more conservative hypothesis, a more stringent, more strict hypothesis, because I really require that there's more signal as compared to the background, as opposed to the more liberal self-contained hypothesis. It becomes clear that that I reject the self-contained null already if there is a single differentially expressed gene in my gene set, and that is of course often the case if I have a lot of differential expression in my background. Katri et al. Took in 2012 in their paper here took a little bit a different approach, but dividing existing methods more along like the timeline of development. Basically, pointing out here's three main generation of enrichment methods, with the first generation being centered around this classical overrepresentation analysis, where I basically have first my differential expression tests, and then I select differentially expressed genes above a certain threshold, and then test here the overlap between the differentially expressed genes and the genes of my gene set. I test this quantity here, the size of the overlap, based on the hypergeometric distribution of them, sometimes also referred to as Fisher's exact test, or I will denote it a lot as overrepresentation analysis in this workshop. Now, there are at least two things to point out when it comes to this first generation of tests. The first is you implicitly have here a independence assumption. So you basically, by applying this Fisher's exact test here, you're assuming these genes are independently expressed, which we know in many circumstances is likely not true, because we know there are patterns of co-expressions between genes, there are dependencies, there are correlations between genes. On the other hand, I limit here the analysis really to a set of differentially expressed genes. So I have here some thresholding, and I disregard here putatively many genes that do not satisfy this threshold. And both of these aspects are resolved here in the second generation of method. Gene set enrichment analysis is maybe the classical, the original gene set enrichment analysis method is here an example. So first of all, we really work here on all the genes, which we compare between two different phenotypes, typically between two different groups, maybe case and control. And I sort here the genes based on magnitude and direction of expression change. So I have here the full gene vector measured in my experiment. And then for a gene set of interest, I basically test now whether the genes of the gene sets tend to be rather accumulating at the top or the bottom of the vector. So on the one hand side, I really work on all genes. On the other hand, I estimate the significance here by sample permutation. That means I preserve the correlation structure between genes. That is, I do it basically implicitly. Another approach is to explicitly take known interactions between genes into account here in so-called network methods, where I really take a priori known network information, which is of course often incomplete, but I take known mechanistic interactions between genes into account and try to score them within a gene set in order to come up again with an information whether there's an enrichment or a depletion for these kind of interactions. Just to make sure that there's much more to say, much more distinctions that are made, and the field has developed a lot since these papers that I showed, and I only go here briefly over these various points, but just to make sure that we are using the same vocabulary and just to make sure that all these things are out there. There are many more distinctions that can be done. Of course, a lot of different issues are arising just due to the fact on which data type you're working on, just given the fact that many gene set enrichment methods have been developed on microarray data, and now we're working a lot on RNA-seq data. And the question is now, have these methods been adapted, or are they still applied in the same way? And you have here the full range of methods. You have methods that have a specific RNA-seq mode. You have methods that have been specifically developed for RNA-seq, and then you still have all these old school microarray methods that are partly applied to RNA-seq data without validating whether these specific assumptions that these methods are having are also hold on RNA-seq data. Another important distinction is whether I take the direction of the expression change in my gene set into account. So whether I basically test, are the genes in my gene set, do they tend to be together upregulated, or do they tend to be together downregulated? As opposed to methods that are testing a mixed hypothesis, where I basically just ask, is there an enrichment for differential expression disregarding of the direction of the expression change? A lot also has been done in the direction of how to do your experimental design. So traditionally we would compare sample groups, but then of course sometimes you don't have replicates, or you're interested in specific individual samples and you still want to score like activity of a certain gene signature. So you have these class of single sample methods. I will mainly talk here about methods that compare sample groups. And then the question is, how do you do that? Do you just have, like, as for example, the original GSEA method, do you have these binary two group comparison, a case control setup? Or do you have, like, for example, a regression approach for those of you that are familiar with like lemma, edge, or seq, and you are able to accommodate also more extended or more complex experimental designs and include covariates also here in the model. Things that have become more and more popular, given that we're now talk about not only transcriptomic data, but in the multi-omic era, we also generate a lot of times gene lists from other omic experiments, and methods that have really become popular is here to do kind of a pre-ranked enrichment analysis. So you really try to decouple the differential expression analysis from the gene set enrichment analysis, which you would typically do on the full expression matrix. But then there are situations where you don't have maybe this full expression matrix, and you maybe have generated a gene list from another high throughput experiment. And then it is very useful to just, apply gene set enrichment analysis on such a pre-ranked list of genes, maybe accompanied by a gene score. While I understand that this is very useful in practice due to the reasons that I just explained, I just wanted to note that always when you do that, you have implicitly the independence assumption. And as I said, that is most likely not true. And while I understand that in many circumstances, this is still a fast and effective way to do it, and maybe sometimes without alternatives, I would say if you have the full expression matrix available, I would always recommend to do that. Yesterday in Raphael Irizarry's talk, we also heard about the importance of exploratory data analysis. So if you have the full expression matrix available, you can always go back and check on individual differential expression and look in more detail in what's going on in regions. Another thing that comes frequently up when I talk in this workshop, I talk a lot about methods, I talk a lot about overrepresentation analysis, where I talk a lot about gene set enrichment analysis, and then often a cause of confusion is that people ask, so what about David, or what about cluster profiler, or what about enricher? And to me, these are enrichment tools. So an enrichment tool implements one or more enrichment methods, allows the application of such methods on different gene set databases, and most likely has some exploration capabilities, visualization of gene sets. And just to bring up here a little bit the table, these are known, well-known or popular enrichment tools such as David here, 20,000 citations, or also enricher and cluster profiler. And basically what I want to point out, they all implement in one way or another a hypergeometric test or the classic gene set enrichment. So that's an important distinction. And I will mainly focus here on the methods, and we keep in mind that these major tools implement these methods. Other important distinctions are many methods typically do sequential gene set testing. So they basically have this uniset approach. They test one gene set after another. But then recently there are also methods that employ multi-set statistics in order to take into account overlaps between gene sets or crosstalks between pathways, so they don't analyze gene sets in isolation, which is maybe a bit more of a realistic perspective, because there are such interactions also between gene sets, not only between individual genes. And that is pretty much linked here also to the last point. Gene set enrichment analysis, you cannot only look at the method, there is always also this influence on what is your database that you're carrying out gene set enrichment analysis. And there are many different databases. I'm just listing here 3. And already these 3, each of them comes with specific characteristics. When I talk about the gene ontology, I of course have these parent-child themes. I have these directed acyclic graph, where I need to take into account these redundancy that is basically implied by the structure of the gene ontology. If I talk about KEC pathways, I need to know there are interactions between these genes, and there's crosstalk between these pathways. And then, for example, if I talk about M60B, I need to understand these are many different collections that are basically representing very different gene sets, again, with very different implications. And many, many more distinctions. So given all these different methods out there, one thing that was really important for us back when I was still doing my PhD back in Munich was to be able to try out many different methods on different gene set databases, and to be able to compare results, or maybe also to combine results and to check whether there is agreement in the enrichment between different methods. And that is basically here this enrichment browser package, 6 years now on Bioconductor, and I will base most of the workshop contents on this package here today. Now, what's this package about? That's a figure here from the original publication in 2016, a bit of a complex figure, but let's concentrate here on the left-hand side for a moment. So this package basically analyzes here these major sources for transcriptomic data. Looking here at RNA-seq data, we have some functionality to basically get this data processed and carry out differential expression analysis with these popular tools for that purpose, ending up with differential expression measures for all the genes that are in my experiment. And then on the other hand, the package basically allows to define gene set according to different gene set databases. Here in the figure, for example, we are able to define gene sets based on the gene ontology or the CAG pathway database. Now, given these two inputs, like my gene sets under investigation and the differential expression for each gene, I'm then able to carry out a number of different set-based enrichment analysis methods. I have here the classical Fisher's exact test and, for example, gene set enrichment analysis. And then the third generation of methods that I'm introduced are basically here featured in another set of functions that are living under the roof of network-based enrichment analysis methods. And for that, I'm not only needing my expression data input, my gene set, but also network information. And for that, it's typically best to really obtain specific organism-specific information, for example, from the Regulon DB, from E. coli, or from yeast tract for yeast. And in cases where such transcriptional regulatory network information is not directly available, we also have some workarounds here to, for example, compile a gene regulatory network from pathway databases by just like compiling all the regulations together that you find in different pathway databases here in the figure, for example, from CAG. And then I'm able here to carry out a number of different network-based enrichment methods. In the end, they all end up with a gene set ranking. So I have here all my gene sets of interest prioritized by a score or a p-value. And then I am able here to either combine or individually inspect these gene sets a little bit further in an HTML report. So that's the basic functionality of the package. That's the original figure. Over the years, we included a couple of more methods for set-based enrichment analysis and also for network-based enrichment analysis. We have here 10 methods each. I could go back to the paper again to maybe say a little bit more about which kind of set-based enrichment analysis methods we have here. So these are methods that are basically covering a decade of developments, quite well established, with the original gene set enrichment analysis paper cited over 16,000 times. And you already see they have quite different approaches in how to calculate the individual gene statistics, quite individual means of how to then summarize the gene statistics on the gene set level. And then they apply for the significance estimation of whether you have an enrichment, yes or no. You either have like the application of specific tests, such as Fisher's exact test for the overrepresentation analysis, or many methods also employ a sample permutation in order to preserve the correlation structure between the genes. So let me bring this back on full screen. So as I've shown, especially given all these popular enrichment tools, David, Enricher, Cluster Profiler, we have a lot of the standard Go CAG overrepresentation analysis in practice. That is what we see most often carried out. We also see a lot of gene set enrichment analysis, and then we have, and that's maybe is sometimes also a cause for confusion. We have a lot of other methods for that purpose that do a specific different approaches and that might be suitable for other specific scenarios. And they are also partly available, implementations in CRAN or in Bioconductor. And then in addition, you have like a constant influx, you have a constant income of additional methods that are developed for specific data types, RNA-seq or single-cell RNA-seq now these days. And the question is a little bit, how do these methods, of course, compare to each other? And that's has been quite difficult to say, because it was, well, you hardly have a chance to really say what are the true positives for a given data set, what are the truly enriched gene sets, and so you were never really able to compare this consistently. And that was basically the motivation for this package that we recently brought out. This is this benchmarking package, which basically collects here a number of well-standardized data sets and allows to carry out all these different methods that are there in the enrichment browser and evaluates the outcome according to a number of defined criteria. So as Levi said in the introduction that just came out beginning of this year in briefing of bioinformatics, and what we did here, what the package does, it basically allows you to efficiently execute all these methods on a compendium of data sets. So we have here 40 different data sets, and you are able to send off, if you're working on a computer cluster or multi-core machine, you are able to send off all the methods on one data set at a time to a specific core that allows you then to assess these 10 different set methods, 10 different network methods. Of course, that is only a representative collection here in the enrichment browser. We have also means to extend this collection, so to define your own enrichment methods and benchmark it, so it's extendable. And then you can look at things like how fast, how applicable are these methods, how many gene sets do they typically return a significant type 1 error rates, and giving some a priori defined relevance ranking. Do these methods indeed like rank gene sets high that we would consider a priori relevant for the phenotype? So for more details, I would then recommend a reading of this paper. Okay, checking on the time. All right. I would maybe say we are also looking a little bit into an example of the application. And for that, I would basically go here to this vignette that, as I pointed out here in the chat, lives under this website. So you go to waldronlab.io and enrich omics, and you basically end up here in this vignette. And on the other hand, I open such a cancer data science instance. So you basically would here click on this link, run the workshop live. Most of you are now already well experienced from yesterday how to do that, but just to make sure everybody is able to do that, username, after you click on this instance and select functional enrichment analysis from the workshop list, username is RStudio, and then welcome to BioC 2020. All right. So if you are in such an instance, you can try out your number of commands. We have here a lot of contents in this workshop. As you see, I will give a little bit an example here on the gene set analysis part. There's more on network analysis and genomic region analysis, which I maybe just give a glance on. Relevant literature here, well, the original publication describing the software is this BMC bioinformatics paper. If you're really interested in how do these different methods in the enrichment browser compare against each other, I would recommend a read of this benchmark paper. Concerning the network contents, there are earlier works here describing a method to incorporate network knowledge into gene set enrichment analysis. and also a paper that basically defines basic requirements that you need for gene regulatory network information in gene set enrichment analysis. Okay, but I wanted to show a little bit how this works in practice. And for that, I would basically consider here a popular example data set in bioconductor. It's the airway data set. Maybe you have already used it yesterday in one or the other workshops. If not, here we are basically comparing transcriptome profiles of different smooth muscle cell lines, which are either or not treated with dexamethasone, a drug which has received a lot of attention lately, given that it's apparently also effective against COVID-19 infection. And what I would do is I would basically load here this data set into my session, and then I would basically subset this data set to only ensemble genes. And if I look here into the assay data, I see I have here read counts for the individual cell lines over my different ensemble gene identifiers. Now, the first thing that I would do, I said we're talking about a differential expression setup. So the first thing that I would do, I would annotate a group variable that would basically, divide my samples into those that are treated with dexamethasone. So these are my case group, as opposed to my control group, which is the untreated group. And I also take here into account the cell line. So these samples are paired, and I find this here more generally in a blocking variable. So I say also take into account the pairing of the samples by taking here the cell line information. Now, the first thing that I would do, maybe in order to carry out an overrepresentation analysis, I would carry out differential gene expression analysis. Given this summarized experiment that stores me here RNA-seq data, and I'm able to carry out here different differential expression methods, including lemma, together with the Bloom transformation for RNA-seq data, or EDGE or DC. Here we are using EDGE, and then we are basically here excluding a lot of genes that are not satisfying minimum expression threshold. That's an argument to the function. You could turn that off if you don't want that, but it's typically a good idea to do that to basically exclude non-informative genes. And then we would find the information of differential expression annotated to the raw data slot of my summarized experiment. At the least, this contains the fold change for each gene and, a p-value, which is adjusted for multiple testing correction. Here, Benjamin Hochberg, but that's an argument to the function. Okay, that's my starting point. And then, of course, in order to perform gene set enrichment analysis, I need gene sets. And for that, we have here this get gene sets function in the enrichment browser, where given a certain organism here in 3-letter CAG code that is Homo sapiens, and I define here basically gene sets based on the database CAG. And if I do that, I'm getting here a cached version, and then I'm looking into how do the first 2 gene sets look like, how many are there in total. In total, there are 337 gene sets. The first 2 describe glycolysis and TCA cycle, and you have here basically the Andre gene identifiers annotated to it as a character vector. Another thing would be to maybe define gene sets according to the gene ontology, where we know in the gene ontology is always important to say, are we talking here about biological process, BP, or cellular components, CC, or molecular function, MF. Here we use BP for biological process. And then I would also have like Go gene sets, again, in the same format. We have here the identifier and the title of the gene set, and the Andre gene identifier for these genes. I could also define my own gene sets or gene sets from other sources if I use the GMT file format here, using an example of file which is available in the package. and then I can call get gene sets on the GMT file. Functionality that I added lately is that we can also define gene sets to msicDB. msicDB defines gene set, as we said, in different collections. And I could, for example, say I want here these hallmark gene sets, the H selection, then I would say here category H. And the good thing is I have here this available for 11 different species. Here I use Homo sapiens again, and then I would get here the hallmark gene sets. The Enricher tool has become quite popular just due to the fact that it has a collection of many additional gene sets, such as transcription factor binding sites and others. You see here, you have here many different gene set collections that are defined on all kinds of different things, ENCODE, human gene and atlas, and so on and so forth. And here the get gene sets, I could also use database enricher and then define one of these libraries. Here I would, for example, use these transcription factor PPI library. And if I do that, I could also then look here into the gene sets, and then I would have here the first two transcription factors and all their protein-protein interacting partners. So there's quite some functionality to define gene sets here, depending on different organisms and different databases. And then per default, this all comes back as Entrez gene identifiers to be compatible with downstream functionality in the enrichment browser. But if I, for example, wanted to have this not on Entrez gene ID, but maybe on ensemble gene ID or human readable gene symbols, I could always map that using here this ID map function. Here I would, for example, map these gene sets to symbols, and then I would accordingly have here the different gene systems. Okay, one thing that I wanted to show is how to actually then carry out a gene set analysis. I can also map my data set from ensemble to Entrez gene identifier just to make sure that my expression data and my gene sets are on the same base. If I do that, I, in the end up, if I look again at the assay data, I then see, okay, now this has been mapped from ensemble to Entrez gene identifier, and then I really can carry out here my overrepresentation analysis. How do I do that? Well, I said you need your expression data and you need your gene sets. This is here just an example, but we could, as we do this live, we could do a real example. So let's say we use here the Go gene sets. And this is like the classical overrepresentation analysis. So this is my Fisher's exact test. I do not do any sample permutation here. That's why I said firm equals 0. And what comes back is a very general table which stores each gene set together with p-value, which is basically calculated based on the hypergeometric distribution, where I have 71 significant genes out of 129 genes in total for this gene set. And I could basically look here into how the ranking looks here for my Go gene set. And that would basically give me here, let's have this in a way that we can see the names better. Let's check here the top 5 gene sets. So we have here, for example, extracellular matrix organization. So maybe the first thing that I would then do, this is here the top enriched pathway. The first thing that I would then maybe do is to look up extracellular matrix organization.
Just to give you a heads up, Ludwig, you have lots of interesting questions, so you might want to make sure to leave some time at the end for questions.
Perfect. I take 1 more minute to finish this example, and then we go over to the question. DEXA matter zone. And then I would basically check a little bit on what is here available. Maybe if I do that, I would maybe have a little bit more of an idea of what dexamethasone actually does and how does it actually influence the extracellular matrix organization. There seems to be a link here given a couple of articles. And of course, this gene set enrichment analysis would be just the start. The last thing that I maybe point out before we go over to the question part of the workshop is that you could then not only see here this flat ranking, but we have, that's why it's called enrichment browser, we have a lot of functionality to then explore these gene sets. And in order to do this, to create such an HTML report, the most important function is here this EA browse function, which I would give such a gene set enrichment result object. And in order to speed this up, I say, just show me the first three gene sets here, the top enriched 3 gene sets. And then it basically would here create such a report for me. And if all works well, it would open here such a result table for me, which I could then use to inspect these 3 gene sets. Let's order them by p-value. Extracellular matrix organization was the most significant one. And then I could look into this gene set here, extracellular matrix organization in more detail. See here a volcano plot, fold change and p-value of the individual genes in this gene set. We see here, for example, this gene is very interesting, has a high fold change and a significant p-value. And then I could look more detailedly here into what's actually with these genes by basically scrolling over them. And I also have a heat map representation of the gene sets, of all the genes in the gene sets, and then also only those that satisfy thresholds for differential expression. So that's very handy functionality to explore gene sets further. I think with that, I would really go over to the question part, just pointing out that this SPEA function, as we just used it to carry out overrepresentation analysis in exactly the same way, providing your expression data set, providing your gene sets. You can then also apply other methods, such as, for example, GSEA, given your expression data set and your gene sets, or for example, those. All right.
You have about a dozen questions here, which I'll take as a sign of interest, not of confusion. Most of them do seem to be of interest more than confusion, but maybe what I will do is any that we don't get to now, I'll post them in the BioC 2020 workshop Slack channel, and you can answer some of them later. The most popular question was, is there a best practices for single-cell RNA-seq enrichment analysis, and what would be the important points and tips that we should pay close attention to if using those methods for single-cell RNA-seq, if not bulk?
Yeah, that's a great question. That is something which I maybe didn't point out here at this point. I mean, here we were talking a lot about bulk analysis, and the cool thing about single-cell RNA-seq data analysis is, of course, we have here now really additional questions that we can try to answer with single-cell RNA-seq analysis, for example, the cell type identification and trajectory analysis. But in the end, I again also there want to do, of course, gene set enrichment to identify cell types, or maybe do gene set enrichment, what genes tend to be enriched, like when I look over the trajectory. So that is, of course, a field of a lot of active development. I think my best guess here, what I would do is to look at orchestrating single cell analysis with bioconductor, which has a specific section also on gene set analysis. And it lists a number of packages which have been specifically developed for a gene set enrichment analysis on single cell data. I think one thing that is used often these days is this ROC cell package here in Bioconductor, maybe a first good starting point, which really applies applies gene set enrichment analysis by taking into account specifics of single cell data. On the other hand, I mean, one thing that you really want to do here is a variance stabilizing transformation. If you are doing something like gene set enrichment analysis, I was skipping a little bit over that, but one thing that you would really do here in order to apply some of the legacy methods would be to stabilize the variance in order to have a data distribution that looks more like what these legacy methods are used to see. But then I really would like to point out that this is a very new field, gene set enrichment analysis on single cell data. I myself work on this currently, and I think we see here also a lot of developments coming up in the future.
Thank you. Is it recommended to use the Reactome database?
Yeah, why not? I think it's a great source for pathway enrichment analysis, especially now that CAG is more and more restricted. I think it has become quite a standard, and the good thing is I didn't talk a lot about network enrichment analysis, but we have here this compile GRN function, which basically allows you to compile a network based on pathway data information of a specific database. And I think if you look into the help of this enrichment browser function, it would allow you a selection of quite different databases. For some reason, looking up the help is here a little bit slower than usual, but if you wanted to see what kind of databases are currently here supported, you see, let me maybe open this here, then we see this better. Compile GRN.
While you're doing that, I'll put the next two questions to you and combine them into one, which is, does your benchmarking workflow give a summary of the overlap or differences between different methods, and can it take into account covariates, such as gene length? Actually, that's not what I thought they meant by covariate. I was thinking of something like age or something about the specimens, but some kind of covariate.
Right. Shortly pointing out this Reactome question, how I finally managed to pull up the help of the compile GRN function, going back to the terminal. When everything breaks down, you go back to the terminal. And then you could basically also define here your pathway database as Reactome, and it's of course as valid as also using other databases such as Biocarta or the NCBI database. So far to this question, the other question that you just said is, can I give a summary of the overlap or difference? It does a lot of different things. I think one thing that it talks about is how do these methods quantitatively compare to each other? So we are looking on the one hand side, how do these methods, here we have these different methods, we are looking, when I apply these to 40 different data sets, what is the typical runtime? So what are methods that are working quite fast? What are methods that are taking quite long? So we are seeing here, for example, the gene set enrichment analysis takes, when I analyze Go gene sets, so 5,000 gene sets over 42 data sets per data set, it takes around 15 minutes, whereas here are other methods that take only a couple of seconds. So I look into runtime. Then I also look, for example, how many gene sets do these methods actually return as significant? If I say significant is an FDR below 0.05, then I see some methods, these competitive ones, they are very conservative. often enough do not return a single gene set as significant. And then there are these self-contained methods that are sometimes overly liberal and tend to return all the gene sets that you test, so 100% of your gene set as significant. It looks into type 1 error rate and also how do these methods behave on random gene sets. You see that some methods, when just your gene set size increases, you get significant gene sets anyhow. And then it also tries to evaluate, do these methods actually rank gene sets high, which I expect to be relevant for a specific phenotype. Here we use like predefined relevance ranking, where we would say, okay, these gene sets are relevant for this specific phenotype investigated in this data set. And we basically summarize this information in a relevance score. And you see here are some methods, this PADOC method, for example, which tends to return highly relevant gene sets for the phenotype investigated. That is what the method does. It gives a quantitative comparison of the method, but then, and I think this is maybe important for how to apply gene set in practice, it talks a lot about like, what do we recommend to do when you work with simple gene lists, and compared to gene lists where you maybe have also a score, how to provide your data if you work on the full expression matrix, if you work with microarray RNA-seq data, and how to deal, and I think this was where the question was going, how to deal with more extended experimental designs when I have covariates. And then I would here also give specific recommendations which methods are very well suited for this purpose or do allow that. I mean, once again, some methods do not allow that. They are just like implemented for basic case control comparisons, and then it is not possible in these tests. So for all these kind of things, this paper will be the reference.
I think the question may have also been actually targeted towards enrichment browser and for analysis, whether you can summarize overlap and differences and use covariates, or if you need to go to the original. Well, the first one, I think I know the answer, whether enrichment browser can summarize overlap and differences.
Right, there is actually a specific function for that. That is something that is here. If you go a little bit more down, there is this combine results function. And you could basically define here a list of different enrichment analysis results. Here I have an overrepresentation analysis result and a result from a gene set enrichment analysis. And then I could basically say, okay, combine here the results, and that would give me here such a combined ranking that gives the relative rank. So this goes from 1 to 100. So 1 is the best, and then it would compare as a summarized measure the mean rank from the two methods, and those that have here a low mean rank means these are the gene sets that are ranked highly by both methods here or by several methods under investigation. And again, that is an enrichment analysis result object. You could call EA browse on this object. I think that is the first part of the question. And I think the second question is, to which extent can you define your experimental design in the enrichment browser? And I think the main functionality is supported. In order to have here a unified interface, you basically need to find here the smallest common denominator. And what is supported by most of the method is a binary grouping, and additionally, optionally accompanied by a blocking factor.
All right, next question. Well, I don't know if we're allowed to go right up until the hour. Maybe we can go a little bit more and people can just drop off. So can you offer advice or best practices for the poor people using studying non-human genomes, such as zebrafish, where the homologies can be quite distant and many to many?
I think this is...
I'll add that Ludwig's first postdoc was studying cattle, so it's something that he is aware of.
Yeah, indeed. I worked a lot in cattle and I worked a lot in yeast, so I know well what you're talking here about. Just pointing out that in this interface, as long as you're working with a organism that is, for example, supported in CAG or Go, I mean, if we go here at CAG genomes, CAG organism, this is, I think, what I want to have. You basically have here this list of 3 letter codes and what kind of organism you're talking about. And you have here quite some organisms. And if you wanted to, for example, let's say we want to get the gene sets from CAG, which are defined where the organism is Saccharomyces cerevisiae. So yeast, you could still use dbCAG, for example. Let's say these are here our gene set. Well, you would need to say here CAG. I have here 1 to 9. Let me bring up this console here. Get gene sets. You could just change basically here the organism of your interest, db equals CAG, and you would get your gene sets as well here for Saccharomyces cerevisiae. I think the question was zebrafish. We could check here what's zebrafish. Zebrafish.
And are there any things to watch out for, gotchas or?
I wouldn't say so. I wouldn't say so. I mean, you get the gene set definitions here by just using DRE. You would also get your network for that. I mean, zebrafish is a quite good animal, actually. It's a model organism. It has a specific genome database, so you get your high quality database. It gets difficult. For example, I was dealing with this Brazilian cattle species, Bos indicus, and there you basically have all the information mapped from Bos taurus, like the general cattle, and then you get in all kind of trouble.
All right, thank you, Ludwig. I think we are out of time. So what I'm going to suggest, there's a whole bunch of other interesting questions there that we take those and create a thread in the BioC 2020 workshop Slack channel to answer them.
Absolutely, absolutely. Well, thanks for all these great questions.
All right, thank you, Ludwig.